Hello, this is Ed Chapman, and this video cast is going to attempt to do a really quick review of what we've been talking about for actually a pretty long time, and what we have also tried to demonstrate in a couple different labs. And this idea is the idea of crossing the membrane. And when we talk about crossing the membrane, we're always talking about something called the plasma membrane. All right, the plasma membrane is what they used to call the cell membrane that separates the cytosol of all living cells from the environment on the outside. All right. And of course, there's some differences between plant and animal and bacteria and all those things. But all cells basically have the same idea. They have stuff on the inside, okay, and they have stuff on the outside. And the stuff on the inside is different from the stuff on the outside. And there is traffic of material into and out of the cell. And this traffic is highly regulated. All right, and it's the regulation of this that keeps cells alive. Okay, it's a big part of homeostasis. Now, there are 5 different ways that we focused on by which material can enter or leave a living cell. Some of these the cell can control and some it can't. Okay, and some types of cells are better at controlling this than others, depending on the environments they've evolved to live in. Diffusion, osmosis, and facilitated diffusion. Okay, everything on this side of the line, okay, over here, everything this way is free. Okay, it requires no energy needed. And the reason for this is, the reason why it doesn't require any energy is because material is always moving down a concentration gradient. All right? Material is flowing in a direction that it normally would flow just because of the laws of physics. Okay, molecules are always going to move from where there's a lot of them to where there's not very many. And because the direction is down their concentration gradient, it's free. It doesn't require any energy. It just happens spontaneously. Active transport and bulk transport, on the other hand, these do require energy. I don't know why I made that a G. All right, they do require energy, and this energy is almost always coming from ATP. All right, so these don't happen for free. So a cell may need to pump material, which is active transport, or swallow or disgorge material to perform its job, and to do that, it's going to need energy to make that happen. All right. Now, diffusion is how most cells get their oxygen and their carbon dioxide. It just soaks in or it soaks out, depending on where there's more of it. Osmosis is a special kind of diffusion. It's the movement of water across a membrane, okay, but the membrane is semi-permeable, meaning that it blocks some solutes. Because there's a difference in solute concentrations from the inside to the outside, we develop something called a water potential, or a difference, or the potential of water to move. And that's what's controlling osmosis. We'll look at that in a little bit more detail in a minute. Facilitated diffusion is the movement of molecules through special pores or special embedded protein gates through which they otherwise, these are openings in the plasma membrane that are necessary for these molecules to pass through, either because they're too big, like glucose, to get through the bilayer, or because the material just needs to move more quickly. Aquaporins, of course, speed up the diffusion of water molecules, or the osmosis of water molecules. Active transport we talked about primarily in 2 different ways. First, we looked at the sodium-potassium pump, which is a mechanism by which an embedded protein can pump sodium ions out of a cell at the same time that it pumps potassium ions into the cell. And we also talked about the pumping of hydrogen ions, again, powered by electrons flowing through an electron transport chain. We saw this in mitochondria and in chloroplasts. And finally, in bulk transport, we talked about certain cells that swallow other bacteria for food or as part of an immune response, like phagocytes, which are a type of white blood cell, or the exocytosis of vesicles, which are produced by the ER and the Golgi, and which work their way to the inside surface of the plasma membrane, join with it, and then disgorge their material to the environment or secrete their material outward by literally turning themselves inside out. All right, now in order to show you guys diffusion, we did an agar cube lab with those pink cubes of agar. And we stressed the importance of a ratio called surface area to volume. And we realized that diffusion sometimes is too slow to move material into the center of a cell. And very simply, we set up a situation where you have a cube okay, with a certain surface area, which you could measure by figuring out how much area you have here, how much area you have here, how much area you have here. And then we also figured out its volume, okay, which was its height multiplied by its width multiplied by its depth going this way, all right? And we realize that as a cell gets larger, as cells get larger or as cubes get larger, the surface area to volume ratio goes down. Okay, and we know that because volume increases a lot faster than surface area. It's just mathematics here. And we realize that when we put different size cells in vinegar solution and cause them to change color, the little cells, we pulled it out when the little cell went completely clear. All right, now I just colored that in, meaning it went completely clear. That doesn't make sense, so let me get rid of that. So we put some cells, 3 different size cells, in vinegar, and we noticed that the middle part of the larger one and a smaller area of this one stayed pink, but the smallest one had enough time to turn completely clear. And what that showed is that if you just rely on the slow effect of diffusion, larger cells just don't have time to service or deal with materials in their centers, okay, because diffusion is just too slow. Excuse me. Now, osmosis is also slow, and it's also free, but it deals primarily with the movement of water. And to talk about osmosis, you have to talk about comparing what's on one side of the membrane to what's on the other side of the membrane. All right. So the basic problem in osmosis, you have to compare what's out here to what's in here. So we do that using these words, these hypotonic, hypertonic, and isotonic words, all right? And we'll talk more about what they mean in just a second. And to show you guys this, we made little model cells out of dialysis tubing, and we quantified a value called water potential. Remember that symbol, the Greek letter psi. All right, so for the dialysis tubing lab, we could reenact it really quickly here. All right, you could make a little model cell, put it in here, make another model cell, put in here, repeat this 2 more times. All right, and let's go ahead and say that this model cell that we put in these 4 different environments contains 0.4 molar solution of sucrose. Okay, now we know sucrose can't get through the dialysis tubing because it's too big. Large molecules are more easily impeded by membranes than smaller molecules. It's just a size thing. Big things have trouble fitting through holes. Small things can go through openings better. All right. So if all these cells are the same, then these 2 environments here have a lower concentration of solute than the cell. Because remember, the cell is 0.4, this is 0, and this is 0.2. So we call these types of environments hypotonic environments. All right, this one is balanced, so that's an isotonic environment, and this environment here is much higher than what's inside the cell, so this is a hypertonic environment. All right, and again, we're talking about the environments, okay, compared to the cytoplasm of the cells. So whenever you use these words hypo, iso, and hypertonic, you have to tell the reader or tell the teacher when you're using this about what you're talking about, the environment or the cytoplasm of the cell. Now, water potential, which we use that symbol psi for, okay, is a way of quantifying this. And you got a little formula that the psi value, the psi or the water potential caused by the solute is equal to negative little I C R T. All right, and what this means is the lower the water potential, okay, the more likely osmosis is to move water towards it. It's as simple as that. So the more negative this number gets, okay, the more likely it is to attract water molecules. That's basically what's happening here. And these all mean different things. Of course, this is the ionization constant, okay. This is the concentration of the solute in moles. Okay, that's the one we're most interested in. This is a constant, pressure constant, and this here is temperature. All right, you'll always be given this material. So let's go back and look at this lab now. Now, what is the psi of these environments? All right, so the psi value for this one is negative little I C R T. All right, the distilled water has no solute, so that means the concentration is going to be equal to 0. So the psi value here is going to be 0. All right, psi value for this one, okay, assuming everything else is constant, is going to be negative little I 0.2 RT. We don't worry about it. So we can say this one is the psi value is equal to 0.2. All right, we're just going to ignore these other factors, or negative 0.2. And this one, again, if you plug it in, negative 0.4, and this one, negative 1.0. All right, excuse me there, that's kind of sloppy. But I think you guys get the idea. As you increase the concentration of the solute, you decrease the psi value. And it's the psi value, the water potential, produced by the solute. All right? And remember, water is always moving towards the lower psi value. So as we saw, when it gets to 1.0 molar, higher than 0.4, which we had inside our little model cells earlier, the psi value, or the water potential in the environment, is lower than what's inside the cell. So then we switched and we saw water starting to move out of the model cell. All right, another way of thinking about this, okay, is with that graph. All right, so if we graph, okay, this is our zero value. If we graph here on the y-axis, percent change in mass over time, okay, and you have a cell that increases in mass, it gets larger, and then finally it can't get any bigger and it levels off. Of course, it wouldn't have to, this would be like a dialysis tubing cell that has absorbed all the water it possibly can. We would call this type of cell experiencing a hypertonic environment. Wait a minute. Oh, wait a minute, did it wrong. Hypotonic environment. Okay, notice here it's environment, because hypotonic environments cause cells to swell, to get bigger, whereas a cell that's experienced a hypertonic environment is going to lose mass before it levels off. So this is a hypertonic environment, and one that's completely not changing is going to be the isotonic environment. All right, so hoping this helped you guys combine the ideas that you were hopefully able to actually observe in these two model cell labs that we did. Thanks for listening, and we'll stop there.